Hi, I'm Kate Ihle, the new molecular biologist at the lab. Thanks for joining us. And today I'm going to be giving a brief overview of the current breeding efforts we have going on here at the lab, along with partners and collaborators across the country. Honeybees in the United States are kept in all kinds of environments and at scales ranging from hobbyists with a few colonies to commercial operations with tens of thousands of colonies. The majority of colonies are part of commercial beekeeping operations. And like a lot of other systems in agriculture in the United States, commercial beekeeping happens at a big scale, which itself can create new challenges for honeybees. Here, I've illustrated with a few pictures to show some of the important sources of stress for honeybees right now. First is a semi-load of bees being ready for transport. The necessity of moving bees across the country to provide pollination services means that they need to deal with adjusting to new environments and even just general stress from being moved and disturbed. The second picture is of colonies in an almond orchard prior to the bloom. And you can see how the colonies are tightly packed, there's so many of them, and how limited the floral resources that are available. The beekeepers in this picture are in the middle of doing a supplemental feeding of sugar syrup and pollen substitutes. And these are really stopgap measures that don't necessarily provide the bees with high quality nutrition like they would get through their own foraging efforts. The next photo is one of the bees' most famous stressors, the varroa mite. And these mites were introduced to honeybees from another apis species, the eastern honeybee. And Apis mellifera bees in colonies left untreated with varroa infestations will die within 2 to 3 years typically. And one of the reasons that varroa infestations are so debilitating for honeybees is that the mites vector several honeybee viruses, such as the deformed wing virus illustrated in the next picture. And as you can imagine, this bee isn't going to be able to forage or to make up for the resources that the colony invested in her development and production. And finally, this last picture illustrates two other major sources of stress for the modern agricultural system: vast monocultures and spraying of agrochemicals. All of these issues have combined, resulting in big colony losses every year as bees struggle to adapt to this new ecosystem that we've created for them. So helping facilitate their adaptation to that ecosystem is the mission of the USDA Bee Lab here in Baton Rouge. We've just celebrated our 90th anniversary at the lab last year. And in that year, they were able to hire 3 new scientists with really diverse backgrounds and expertise to continue the lab's mission. And like I mentioned, I'm the newly hired molecular biologist, and I'm developing a research and breeding program building from my past work that focused on behavioral genetics and genomics. And I'm really excited about the potential of our new programs targeting new traits and generally breeding for more resilient bees. So probably most of you here are familiar with the structure of honeybee colonies, but for those who aren't, honeybees are eusocial animals that live in really big colonies. Most of the members of the colony are sterile female workers. A single queen is responsible for all of the egg laying, and she'll mate early in her life with several drone bees, the males. And the purpose of those males is really only to mate. They really don't do anything else at all around the colony. And this creates a special set of challenges for selection and breeding, because usually we're going to be trying to select for either traits at the colony level, like honey production or mite resistance. And those traits are going to be the result of contributions from thousands of the worker bees. But for us, we have to use the queens and the drones for breeding. So very rarely are we going to be selecting on traits directly expressed by those reproductive queens and drones. Instead, we have to wait for the workers to be produced by those colonies or for the colonies themselves to mature to see how our breeding worked out for each generation. But that's being said, honeybees are really flexible and very amenable to breeding efforts. Two of the most successful efforts in the lab over the years have been the kind of varroa sensitive hygiene, Pauline Ihle breeding program. and the importation and propagation of Russian honeybees, first by the lab and now by the Russian Honeybee Breeders Association. So pollen honeybees were bred by the USDA lab here in Baton Rouge for varroa resistance by selecting for a trait called suppression of mite reproduction. To score this phenotype, someone has to dissect hundreds of nearly mature brood cells to note the presence of varroa mite foundresses, whether or not they've reproduced. It's extremely time-consuming and tedious. Garrett Dodds will be talking about them in more detail later today, and he can tell you all about it. Russian honeybees, on the other hand, are maintained by the lab and were originally imported from the Primorsky region of Far East Russia. This is the region in which Apis mellifera is thought to have had the longest association with varroa mites. It butts right up against the natural ranges of varroa and their original host, Apis cerana. And because of this, their resistant traits are largely naturally selected, and we're still exploring what makes these bees resistant to varroa infestations. It's likely to be a mix of several traits. And Lilia de Guzman at the lab is working with the Russian Breeders Association to keep these Russian honeybee lineages genetically diverse, but recognizably Russian. Lainey Bildo and Lorraine Beeman work really hard on a set of genetic markers that can tell the breeders the degree of Russianness, it's called, the genetic Russianness of their bees. And they then use that information for the breeders when creating the next generation to keep the Russian honeybee genetics from getting too dilute in the general population. And it's been a really successful partnership over the years. So for the next few minutes, I'm going to take you through some of our newer efforts to target novel traits. The first is related to my favorite gene, vitellogenin. And I think I've told a few of you that my husband just is constantly asking me, oh, what doesn't this gene do? Because it's the primary energy storage protein for honeybees. It's incorporated into the brood food. It can protect against things like oxidative damage. It might have a role in this process called transgenerational immunity, which is when immune function is passed on from the queens to her daughters. It affects pollen versus nectar collection. It has a role in this age-associated progression from nursing to foraging tasks. It affects longevity. And more recently, it's been described as a biomarker for colony health. Another thing we do know is that there is genetic variation for the production of this protein, because in selected lines of honeybees bred for high and low pollen hoarding by Rob Page and Kim Fondrick, we see consistent differences in expression of vitellogenin and the levels of the protein in the hemolymph. So we know that there's a genetic component, suggesting that it should be relatively easy to target with a direct selection program for increased vitellogenin production. So what I have planned and begun a phenotype screening for is a breeding regime that will use bidirectional selection for high versus low vitellogenin production. We've collected samples from a variety of colonies and stocks, and we're currently processing some of them, and I'm still conducting some cage studies. But in the spring, I'm going to use that data to begin a breeding program using single drone artificial insemination to reduce that genetic diversity in colonies, where queens typically mate with upwards of 20 or so males, and reduce that down. So I plan to breed divergent lines for 3 to 5 generations, depending on the stability of this trait, and then measure a suite of individual and colony level traits. And these will include things like resilience in the face of moving and other environmental stressors, overwintering success, brood production, and queen rearing. And queen quality is a big concern to beekeepers right now. And a lot of the success of queens depends on their nutritional environment during development. And one thing to note, as biologists, we know that creating these beneficial traits costs a lot in terms of energy. So vitellogenin is this really rich, important protein, but it takes a lot of energy to produce. And we see that there's a lot of variation for this. So potentially there's a cost. So, maybe producing this vitellogenin is great for some characteristics of bees, but really bad for others. And that's something we're really aware of and working on. But one question is also that if there are trade-offs for producing, can we mitigate these by management practices? And would this even matter for some specialized use, such as commercial queen and drone production, where high vitellogenin workers could potentially use all that protein to raise really high quality queens? And this is possible because we're able to separate the genetics of bees that rear new queens from those queens themselves. So queen breeders and rearers raise hundreds of queens in the same colony called cell builders. And the cell builder colonies are these really strong colonies full of young nurse-aged bees. The breeders would then collect newly hatched tiny female larvae produced by a breeder queen and graft them into these containers called queen cups. They're lined up on bars in the same orientation as naturally occurring queen cells and placed into the cell builder colonies. So we can, in that way, select for queens for producing colony traits like mite resistance or honey production, but rear those queens in colonies selected for vitellogenin or even just grow up jelly production. And beekeepers have been reporting that queen quality and queen health have become big concerns over recent years. And because the developmental environment is so important for future health and fecundity of queens, I think this approach could be really promising. This vitellogenin work could also play into a second and related program that I'm working on, led by another new scientist in the lab, Vincent Rosigliano. And this tackles the issue of nutrient deficiency that bees often face when moved or when they're located in a huge monoculture, or when they have to compete with hundreds or thousands of other colonies for limited resources. And we're taking a tack that has been used in many domesticated species, breeding for highly efficient nutrient assimilation. That is, we're looking for genetic variation in a bee's ability to turn energy from pollen and nectar to fats and proteins to new brood and bees. So right now, the first screening steps for this trait is being done in concert with the vitellogenin selection. And we're hoping that by selecting for increased nutrient processing, we could potentially breeding bees with higher vitellogenin production, while potentially limiting these life history trade-offs that we might expect to see. We have some preliminary data here from our screening, and this data is from 6 colonies of 2 different stocks, the pollen and Russian bees that I talked a little bit about earlier. What we did was to measure food consumption of small groups of workers kept in these little cages in the lab. And then we took various measures to tell us how that consumption related to muscle mass, vitellogenin production, lipid, fat content. And as we process the samples for our more fine scale measurements, we're really encouraged to see a lot of variation in these more basic broad measurements. So here we have thorax weights of workers from 6 colonies of each stock after a week in the cages. And you can see that there's a lot of variation in this simple measure between the stocks, but also within the 6 colonies from each stock. And this is data, more recent data from Vincent and Tyler Williams, showing relative vitellogenin expression corrected for pollen consumption. So basically trying to measure how efficiently bees are able to turn that protein that they eat into vitellogenin. And this suggests that there's a lot of variation present in even this really narrow sample of colonies for us to select on. So I'm really excited to see how the more detailed measures correlate with each other and how it looks overall, and hopefully to begin a selection program in the spring. And the last thing that I'm going to cover is a really exciting project led by Mike Simone-Finstrom at the lab and Marla Spivak at the University of Minnesota. And their goal is to combine knowledge about honeybees' natural defenses along with integrative pest management strategies to identify and breed for multiple resistance traits to increase the sustainability of beekeeping and honeybee management practices and honeybee populations. They currently have programs underway to breed for the collection of propolis, for hygienic behavior, and low mite reproduction. Propolis itself is a resinous material that bees in the wild would use to create a protective envelope around their nest. Propolis has antimicrobial properties that might help to improve individual honeybee resistance, immunity, and longevity, which would ultimately lead to better colony health and productivity. And they're combining these efforts with a cool new idea in management practices using these grooved and roughened Langstroth hive bodies to encourage propolis deposition. And so this is a really exciting breeding program that's just getting off the ground, and I think we're going to see really exciting results from it. And all of these efforts that I just talked about are ultimately to help honeybees increase their resilience in the face of the multiple and interacting stressors that they're facing now. And our goal at the lab is not only to breed better bees, but also to use the huge array of skills that we have at the lab, from beekeeping and colony manipulation to insemination and cutting-edge molecular biology and genomic techniques, to dissect both the phenotypes, these traits, and the genetics of those resistance and resilience traits. So because honeybee breeding often requires mature, seasoned colonies for phenotyping, you'd be surprised at how long and tedious the phenotyping can be. It can be a real barrier to large-scale production of resistant and resilient lines of bees. So our big goal in the coming years is to combine our breeding and molecular efforts to make it possible to use genetic or biochemical markers to streamline the selection efforts at the lab and to help our honeybees thrive. And with that, thank you for your time. And if you have any questions, feel free to contact me at kate.ihle@usda.gov. Thanks so much.